Hello students, this is Lohita. So here we are into our zoology NEET class. So we are going to continue to learn regarding the chapter molecular basis of inheritance. Okay, so we are into the chapter molecular basis of inheritance, right? So in that we are going to learn today regarding human genome project. Okay, so we are going to learn about human genome project. Okay. See students, in our previous classes in this chapter molecular basis of inheritance, we have already studied that the nucleotide bases which are present in the DNA are going to form the genetic material. The nucleotide bases which are present in the DNA, nucleotide bases present in the DNA make up the genetic material, isn't it? So, nucleotide base pairs present inside the DNA are our genetic material. This we have learnt in our previous classes, right? So, that means our genetic material is present inside the DNA. So, we are understanding that our genetic material is present inside the DNA. Okay. See students, not all the individuals are same, right? Oh, everybody is different. No two people are same. No two people are similar. Everybody is different in their own way. Everybody have different characters. In different aspects they are different, right? So this is all because the differences in the genetic material, differences in the DNA. Okay. So differences in individuals. Is due to the differences in their DNA, is due to the differences in the DNA. Okay, differences in the individuals is due to differences in their DNA. This is also one more observation that is made by us. See students, with the increasing technology, with the increasing application of different practices, there has been a lot of technology developed to isolate the DNA, to read the sequences of nucleotide base pairs in the DNA, to isolate DNA, to read the sequences of nucleotide base pairs in the DNA. Different techniques have been arisen. So depending upon this technique, keeping these techniques of isolating the DNA, keeping these techniques of isolating the DNA, fragmentation of DNA, cloning of DNA, and also sequencing of DNA, Okay, the technology was developed in all these aspects. Different aspects. We could isolate the DNA, we could fragment the DNA. That means we can break the DNA into pieces, and cloning of the DNA. We'll learn what is cloning again. Cloning of DNA and sequencing of the DNA. Through all these different aspects, technology has been very well developed. After the development of this technology only, an idea for a project has been evolved. Okay, that is human genome project. Okay, so it's a very big project students. Human genome project has been arisen after the development of all these different technologies. Human genome project is a very big project. It's a mega project. It is called as a mega project. It's a very big project which has begun in 1990, which has started in 1990. Human genome project, simply called HGP. It's a mega project which started in 1990. Okay. So in today's class students, we are going to learn about the human genome project. Different aspects related to human genome project we are going to learn in today's class. Okay. So let us learn about human genome project. See students, human genome project, as I have told you, it has started in 1990, right? It started in 1990 and it was a 13-year-old project. It was finished in 2003. Human Genome Project started in 1990 and it was finished in 2003. So, it is a 13-year project. It was a 13-year project. Okay. It was a 13-year project. Human Genome Project, it's a 13-year project. So, students, this 13-year project, Human Genome Project, was ran under the US Department of Energy, US Department of Energy and National Institute of Health, National Institute of Health, under the survivorship, under the supervision of these two departments, US Department of Energy and National Institute of Health. Under these two organizations, this Human Genome Project has started. Later on, Wellcome Trust and some other countries like China, Japan, Korea, okay, some other countries also have joined this project. Some other countries like China, Japan, Korea, these are the other countries which have joined this project later on. Okay. So, students, what is the main intention of this project? What is this project mainly intended to do? What is the aim of this project? Okay, let us learn now. See, students, we have already studied that human genome contains, there are 3 into 10 to the power of 9 billion, 3 into 10 to the power of 9 base pairs, 3 into 10 to the power of 9 base pairs are found in the human DNA, This we have already studied. 3 into 10 to the power of 9 base pairs are found in the human DNA, right. So, students, if we are assuming that each base pair requires $3, sequencing of each base pair requires $3 per base pair, then there are 3 into 10 to the power of 9 base pairs in the human DNA totally. So, for identifying one base pair, if we are spending $3, then for identifying 3 into 10 to the power of 9 base pairs, we require totally 9 billion US dollars. It requires 9 billion US dollars. 9 billion US dollars are required. This much amount of money has to be spent on this project for identifying the entire human nucleotides. Okay, that was a very major issue. And the other thing is, see students, how many are there? 3 into 10 to the power of 9 base pairs are present in the human DNA totally, right. So, if we are using 100 pages book, okay, if we are using 100 pages book and in each page, in one page, if we are adding 1000 base pairs, in one page, then we require nearly 3000 books of such kind. We require nearly 3000 books of such kind. One page, 1000 base pairs, each book having 1000 pages, then we require nearly 3000 books for saving only one human's DNA. For saving only the sequences of only one human's DNA, we require 3000 books with 1000 pages each, in which each page contains 1000 base pairs. In such way, we require 3000 books of such kind for storing the human DNA. Okay, so sequences of the human DNA should be stored in those many books. So, one person requires 3000 books for storing his nucleotide sequences. So, now students, these are all the issues that have to be addressed by the Human Genome Project. So, let us learn what are the goals of the Human Genome Project. Okay. So, let us now learn the goals of the Human Genome Project. The goals of the Human Genome Project is to identify all the 20,000 to 25,000 genes which are present in the human DNA. Okay. To identify the sequences of nearly 3 billion base pairs which are present in the human DNA. Okay. Tools for data analysis, developing tools for data analysis. Okay. Developing techniques for storing of the information, developing techniques for the storage of information. Okay. Application of the results in different areas, application of the results in different areas. Okay. And address all the social, ethical and legal issues which occur during the project. Okay. So, these are the goals of the Human Genome Project. What were the goals of Human Genome Project? These are just goals. Before actually executing the project, before actually working on the project, some goals will be set up, students. So, these were the goals before actually executing Human Genome Project. So, students, to 20,000 to 25,000 genes present in the human DNA have to be known. Sequences of 3 billion base pairs present in the human DNA have to be identified. Tools for data analysis should be done, because we have to identify how we are going to identify this nucleotide bases, that is the tools for data analysis. Storage of information, as we have studied that 3000 books of the 1000 pages each, which 1000 base pairs in each book, then the human DNA's information can be stored. Only for one person these many books are required. So, that storing of the information regarding the human DNA is also a very big task. So, that has to be addressed. Application of results in different areas. See, students, without application the project is waste, right? So, for application of the project into different areas only, we are mainly intending to identify the human DNA, right? So, we have to make out the ways where we can apply this. It can be in industries, it can be in healthcare, agriculture, in different areas it has to be applied. Okay. And address all the social, ethical, legal issues which arise during the project. Okay. So, these are the goals of Human Genome Project. Okay, students. So, by utilizing this Human Genome Project, the DNA of human beings is identified. By 2003, almost all the chromosomal of the human beings, they have been identified. And along with the human, some non-human DNA also has been identified. Okay. Human DNA was sequenced by Human Genome Project, but along with the human DNA, even some plant DNAs was also identified, like for example rice, Arabidopsis was identified. Among animals, among other organisms, the chromosomes of bacteria, yeast and Cenorhabditis, which is a non-parasitic free living nematode. Cenorhabditis, Drosophila. Okay, these were the other organisms whose genome was sequenced, okay, whose nucleotide base pairs were identified. Sequencing of genome is nothing but identifying the nucleotide base pairs. So, these are the ones whose nucleotide base pairs have been identified. Okay. So, let us learn now what were the methods utilized in sequencing the DNA, how exactly the DNA sequences have been identified. Okay. Methodologies in Human Genome Project. Okay, methodologies in Human Genome Project. See, students, in Human Genome Project there were two main approaches. There were two main approaches in Human Genome Project. Okay, The first one was to identify the expressed genes. Okay, first one was to identify the expressed genes. Expressed genes are those genes whose expression can be observed. Okay, they are the ones whose expression can be observed. They are those sequences of DNA which are expressed. Okay, that means they have some proper function. They have some proper function. They are expressed in the human genome. The ones which are expressing themselves, having a proper function, they are called as expressed genes. Okay, these expressed genes are those genes which have a proper function. Okay, they are the ones whose function is expressed, in the human genome. They are called as expressed genes. So, we have to identify what are the sequences of the genes which are expressed. Okay. And next one is sequence annotation. Sequence annotation was the next approach. Sequence annotation means to identify the entire sequences of the human DNA. To identify the entire sequences of the human DNA. Okay. Sequence annotation means to identify all the sequences of human DNA. Okay. All the sequences have to be identified. Okay, all the sequences of the DNA have to be identified. After identifying all the sequences of DNA, we are going to arrange all the sequences. Okay, we are going to arrange all the sequences and see if what are the sequences expressed, what are the sequences which are expressed and what are not expressed. What are the ones which are not expressed have to be identified. Sequence annotation means that only. We have to identify all the sequences first, then after that we are going to see if what are the sequences which are expressed, what are the sequences which are not expressed. Okay, that has to be identified. That was one more approach of Human Genome Project. So, these were the main approaches. So, students, second approach actually covers the first approach already. So, the first approach is to identify only the expressed genes. The expressed genes are those genes which are actually found expressed on the DNA. They are the ones which are expressing themselves. Okay. So, they are having a proper function. Their function is exposed among the genes of the genome. Okay. Then sequence annotation is another function. See, sequence annotation means all the sequences of the human DNA have to be identified. They have to be arranged serially. After arranging them, we are going to see if what are the ones which are expressed, what are the ones which are not expressed, that we have to identify. Okay, and we have to give them a proper tag that these are the expressed sequences, these are the non-expressed sequences. Like that, we have to give them a proper tag. That is the Human Genome Project's two major approaches. Okay. So, let us see now how exactly human genome has been sequenced. Okay. See students, first, as I have already told you, we have enough techniques. Before Human Genome Project was executed only, there were enough techniques for isolating the DNA, for cutting the DNA, for cloning the DNA and for sequencing the DNA. Okay. So, based on all these different techniques, utilizing all these different techniques, Human Genome Project was working. Okay. Let us see exactly how human genome has been studied. Okay. So, the first step in studying the human DNA is isolate the DNA. Okay. So, how do we isolate the DNA? From the cells, okay, we are going to separate the DNA or chromosomes, or if the DNA is present, DNA is separated. If chromosomes are there, then from the chromosome DNA is isolated. If DNA is available directly, then the DNA is taken. From the cells, DNA is taken. Okay, and by utilizing certain enzymes, random fragments of DNA are prepared. Random fragments of DNA are prepared. Okay, by utilizing different enzymes, random fragments of the DNA are prepared. Random fragments, in the sense small pieces of the DNA are prepared. Okay, random fragments of the DNA which are small sized, small fragments of the DNA are prepared. Okay, so this is all like in the steps, students. First we are isolating the DNA from the cells, and after that by utilizing some enzymes we are breaking this DNA into small pieces. Okay, after that what do we do with this DNA is, by utilizing a vector, okay, by utilizing a proper vector, these fragments of DNA, are cloned into a host, okay. By using a vector, suitable vector, the fragments of DNA are cloned into a host. What is this statement means exactly, we have to learn now, because you are not familiar with this cloning or vector. Okay, so let us learn in detail regarding what is vector, what is cloned. See students, we are having only a piece of DNA, right? We are having only a piece of DNA, that is a small fragment of DNA we are having. So we are not going to study the entire human DNA, which is, as we have already studied, that human DNA is going to stretch for few kilometers. So we cannot study the entire kilometer length DNA, right? So for that reason we are breaking it into pieces first, and after that we are using some vectors. Okay, vectors are carriers actually. Carriers. So these are the ones which carry the small fragments of DNA. Okay, these are the ones which carry the small fragments of DNA into a suitable host. Vectors are carrying these fragments of DNA into a suitable host. So these vectors go, take this DNA and they enter into the host. They will join the small fragments of DNA to their DNA. Okay, they will join the small fragments of DNA to the host DNA. Okay, that is cloning. Adding of the vectors, adding of the required fragment of DNA to the host DNA is called as cloning. Okay. So students, so here generally a vector is used. Vectors generally used are YAC, which means yeast artificial chromosomes, BAC, which means bacterial artificial chromosomes, yeast active chromosomes, bacterial artificial chromosomes. These are the ones which are the vectors. Okay, these are the vectors. Yeast artificial chromosomes, bacterial artificial chromosomes are the vectors for cloning of DNA. And what is the host, students? Here host is generally a bacteria or a yeast is the host for this DNA, for this fragments of DNA. See, why are we generally choosing bacteria or yeast? We are choosing bacteria or yeast because they have very small sized DNA. Bacteria or yeast, they are having small sized DNA. Okay. So students, by utilizing these vectors, to these vectors we are first tagging the small fragments of DNA. Okay. To the vectors we tag the small fragments of DNA. Okay. So these vectors carry these small fragments of DNA into the host, where these fragments of DNA, they get amplified, where these fragments of DNA, they get amplified. Amplified means they get very clear, they will be very clear to study. Okay, because human DNA is very long, we won't be able to study it directly. So we are breaking it into fragments using vectors. We are sending it into a proper host who is having very small DNA. In that host we are going to amplify this DNA. If we require, we can produce many different copies of our required fragment of DNA. That is also cloning, producing similar fragments. sequence is cloning. So such similar fragments of the smaller DNA will be produced. We can study the sequences of nucleotides from that similar fragments of small DNA. Okay. So this is how, students, exactly we are amplifying the small fragments of DNA. We are trying to very clearly study the small fragments of DNA by joining them to the host bacterial or yeast DNA, and then we are going to make it very clear and study it. Okay. So this is clear. How we are going to clone it, we have learned now. So we have learned that first we have isolated the DNA from the cells, or from the chromosomes. We are going to isolate the DNA by utilizing some enzymes. Small random fragments of DNA are prepared. Small fragments of DNA are prepared, randomly they are prepared. Okay. And after that, using a proper vector, the fragments of DNA are cloned into a host. Who are the vectors generally? Yeast, YAC, yeast artificial chromosomes, or BAC, bacterial artificial chromosomes, are generally the vectors. Okay. Host is generally a bacteria or a yeast. A bacteria or a yeast is generally the host. Okay. And generally, students, what is exactly happening is, into the vectors the fragments of DNA are tagged, then the vectors are allowed to go into the host, either into the bacteria or yeast, they allowed to go, and these fragments of DNA get amplified in the host, bacteria or the yeast. That means they get very clear, they can be multiplied in the number, they get very clear, very big enough to study. Okay. So now let us see how the sequences of DNA have been identified. Yes, it has got amplified, we have cloned them to the host, they have got amplified. Now how are we going to read them is the task. Okay. So students, according to some principles, based on the principles of sequencing developed by Frederick Sanger, he is the person, okay, who has developed the principles for sequencing the amino acids of the proteins also during translation. Frederick Sanger, students, is the person who sequenced the who has given the sequences of amino acids during translation also, and he is the same person who has given the principles for sequencing of the nucleotides in the human DNA. See students, there are many billions of nucleotides which cannot be readily read by man. For that we have requirement for computer programs. So students, in this level computer programs have been developed which can read the DNA, okay, which can read the DNA. Annotation, we say it as annotation, which are involved in annotation of the sequence, annotation, which is like they are helpful in reading the DNA. Some computer technology has been developed, computer programs have been developed based on the principles of Frederick Sanger, which have helped in sequencing the nucleotides in the DNA. See students, we have obtained only small fragments of DNA, right? Small fragments of DNA ne vache maniki. So whatever sequencing is done in those small fragments only. How are we going to identify the sequences of nucleotides in the entire DNA then? See, based on some overlapping regions. So the fragments of DNA, they have some overlapping regions. They are not cut straight, they are randomly cut. Okay. So if this is the piece of DNA, it is not cut straight like this. It is not cut straight like this. They are randomly cut. They are cut like this. They are cut like this. So here we observe that some of the sequences on the other side are not there, they are randomly cut. No. So we observe that some of the base pairs have to be present on the other side. If there is a G on the other side, there has to be C. If there is C, there has to be G, G, C, like that some kind of clues are left over. So these are the overlapping regions. So these are the overlapping regions. So we observe the overlapping regions, students. We observe the overlapping regions between the DNA sequences, between the fragments of DNA, we will be reading the sequences of the nucleotides using the computer programs designed by Frederick Sanger. So we are going to actually identify the overlapping regions of the DNA fragments. Okay. After identifying the overlapping regions of the DNA fragments, how do we sequence them? For sequencing the overlapping regions of the DNA fragments also, there were some computer based techniques developed by Frederick Sanger, which are helpful in identifying the sequence of these overlapping regions. Overlapping regions ఉన్నాయి, ఓకే అవి ఏ సీక్వెన్స్లో ఉండాలి, ఎలా ఉండాలి, అవి ఏ సీక్వెన్స్లో ఉండాలని ఐడెంటిఫై చేయడానికి కూడా there was some computer programs developed by Frederick Sanger. Okay, based on that computer programs developed by Frederick Sanger, the overlapping DNA fragments have been identified. According to it, the entire length of the DNA fragments can be sequenced. Okay, so in that way overlapping regions were identified and then sequencing was done. Sequencing of the entire DNA could be done. Sequencing of the entire DNA could be done in that way. Okay. So then students, the other problem was, the next problem was genetic mapping and physical maps. Genetic map and physical map. Obtaining the genetic map and physical map were the next task. Okay. So how do we obtain the genetic map and how do we obtain the physical map? So students, genetic mapping means identifying the genetic distance between the nucleotides. Identifying the genetic distance between the genes is a genetic map. Identifying the genetic distance was the genetic map. Okay. That means it is not related to the distance between the genes. It is related between the association of the functions between the genes. Okay. And physical maps, that means how closely or how farly they are located to each other. How close or how far they are located with each other, that is the physical map. So this identifying the genetic map and physical map was the next task. So physical map and genetic map, these were identified by using repetitive sequences and polymorphism of some enzymes called restriction endonucleases. Okay, repetitive sequences, some sequences of nucleotides are repeated by utilizing them, and polymorphism of restriction endonucleases, that means activity of different restriction endonucleases. Based on these factors, genetic mapping or physical mappings were obtained. Okay. So let us now learn what were the salient features of Human Genome Project. What were the observations made after Human Genome Project. Okay. Salient features of Human Genome Project. Human Genome Project identified that there are 3164.7 million base pairs in human DNA. Okay, totally there are 3164.7 million base pairs in the human DNA. Okay. They have also identified that there are nearly 30,000 genes in the human DNA. It was earlier believed that there are nearly 80,000 to 1,40,000 genes in the human DNA. But it's not true. There are actually 30,000 genes present in the human DNA was identified after Human Genome Project. An average gene contains 3,000 base pairs. The longest gene is dystrophin gene. Longest gene is dystrophin gene with its 2.4 million base pairs. With its 2.4 million base pairs, it's the longest gene. Okay. And you know students, nearly 99.9% of the nucleotides are same in all people. Nearly 99.9% of the nucleotides, they are same in all the people. Okay. 50% of the genes function, 50% of the known genes function is not known. 50% of the known genes function is not known to us, is unknown. Okay, 50% of the genes function is unknown to us. There are many repetitive sequences. Many repetitive sequences are present inside the DNA. Many sequences of DNA are repeated. Some sequences are repeated from hundreds of times to thousands of times. Hundreds of times to thousands of times some sequences of DNA are repeated. Okay, hundreds of times to thousands of times the sequences of DNA were repeated. There is no importance of function of these repetitive sequences. The only importance is they help in tracing the evolution, determine the structure, dynamics of the DNA, determine the structure and dynamics of the DNA. Okay, that is the only importance. They help in understanding evolution, structure and dynamics of the DNA. Okay, they help in understanding the determining of the structure, dynamics of the DNA. Scientists have identified nearly 1.4 million sites in the human DNA which were having only single nucleotide difference, differences. Okay, it is called as single nucleotide polymorphisms. Single nucleotide polymorphism. There was only difference in one nucleotide between the different individuals in nearly 1.4 million sites on the human DNA. Okay. So, students, these are the salient features of human DNA. 3,164.7 million base pairs are present in the human DNA. 30,000 genes are present. It is not 80,000 to 1,40,000 genes as we have thought earlier. It is 30,000 genes. An average gene contains 3,000 base pairs. The longest gene is the dystrophin gene with 2.4 million base pairs in it. 99% of the nucleotides are same in all the individuals. 50% of the known genes function is not known to us. Repetitive sequences are found in the DNA. Hundreds to thousands of times these sequences are repeated. They have no importance, they have no function, they have importance in evolution, understanding evolution, understanding the structure and dynamics of the DNA. Scientists have identified during Human Genome Project nearly 1.4 million sites in the human DNA where there are differences in only single nucleotide. Okay. So, between different people they have identified that there are 1.4 sites are found in the DNA which are differing only in one nucleotide. They are called single nucleotide polymorphisms. Okay. So, students are then coming to the applications. Where can this Human Genome Project be applied? See students, by identifying the entire sequence of DNA, we can understand the entire expression of genes in an organ, in a tissue or in the different interactions between the different genes in obtaining a particular function can be identified. Identifying the functions of one gene or identifying the sequences of only one gene at a time may not help in relating with the other aspects, but identifying the entire genome will help us in understanding the functions of organs, tissues, tumors or interactions between the genes in obtaining a particular function can be identified. Okay. Different reasons for the disorders can be identified. Corrections related to the disorders can be done. Okay. But students, this project is still in the developing stages only. This project requires tens of hundreds and thousands of scientists from the different areas to work still on this project to make it more applicable to different aspects. It has applications in agriculture, human health, animal husbandry and different aspects it can be applied. Okay. And one last point that we have to add to the salient features is the longest chromosome is chromosome 1 with 2,369 genes. Okay, 2,369 2968 genes is the longest chromosome, and the Y chromosome with 2031 genes, it is the smallest chromosome. Okay, longest one is 2968 genes in chromosome 1. Y chromosome with only 231 genes, it's the smallest chromosome. Okay. So that's all regarding the human genome project, students. So these are the aspects related to human genome project. So we will continue our class in the next session with we will have to move on to the next concept. We will learn the next concept in the next class. Thank you all for joining the class today. We'll meet in the next class.